is currently the vice chair. He was also recently the chair in the program in dynamical neuroscience at UC Santa Barbara. He has received a Sloan Research Fellowship in Math and a National Science Foundation Career Award, and was program director of the SIAM activity group in dynamical systems from 2008 to 2009. He has supervised 10 students to the completion of a PhD. 4 students received a terminal MS degree, and 3 students received a BSMS degree. Jeff's current research includes applications of dynamical systems and control techniques to neuroscience, cardiac dynamics, and collective behavior. He's published over 100 journal and conference proceedings articles on these and other topics, including shear flow turbulence, microelectromechanical systems, energy harvesting, and dynamical systems of symmetry. We're really excited to have Jeff speak today, and with that, I'll hand it over to you.
All right, thank you very much to the organizers for inviting me. I'm going to share my screen and hopefully this all works okay. All right, so let's see, here we go. Okay, you can see my screen okay, I hope. Oops. Okay, so today I entitled my presentation, Learning How to Control Populations of Neurons. And I'm kind of using learning in 2 senses here. One is, we are learning how to do this. And as you'll see, I'll be presenting some of the older work that got us to where we are today. And also, in the spirit of the theme of this seminar series, as you'll see, we'll do some machine learning for how to control populations of neurons as well. Okay, so I want to start with the motivation, which is Parkinson's disease. So Parkinson's disease affects about 1 million people in the US, including some well-known people. The estimated cost of Parkinson's disease is about $25 billion a year in the US. And this includes treatment costs. It also includes things like lost productivity. The symptoms of Parkinson's, probably the most familiar one is tremors, the shaking of the limbs. But there's other symptoms as well that can be equally or even more debilitating, including a postural instability, slowness of movement, rigidity, things like that. So Parkinson's, there's at least a hypothesis that it's associated with a pathological synchronization of neural activity in the motor control region of the brain. And it's also associated with elevated beta band power. And as you'll see, the control techniques that I'll be considering are to try to combat one or both of these, either the pathological synchronization or the elevated beta band power. Now, the treatment techniques for Parkinson's are either drugs or deep brain stimulation. I'll be focused on deep brain stimulation. And here, a neurosurgeon implants an electrode into the brain of the patient. And there's a wire connecting the electrode to a pacemaker that is implanted in the patient's chest. And the pacemaker generates an electrical signal that's injected into the brain tissue. And if you choose the correct signal, then it can make some of these symptoms go away. And I'm going to show you a video of a patient that had deep brain stimulation implanted. And you'll see kind of, how this works. So this video is an individual named Andrew Johnson. I'll tell you, I was not involved at all with Andrew's treatment. He's an individual that had early onset Parkinson's disease, and he has a little bit of a sense of humor. So he has a blog called youngandshaky.com, and the sound on the video may not come through very well, so I'll kind of narrate it as it happens. But I want to just orient you a little bit about what's going on. So first of all, he's, excuse me, he's going to hold up a little device, a yellow device, that he can use to communicate with the pacemaker, which is implanted in his chest. And at first, he's going to hold it up to his pacemaker to show that the stimulation is on. And then he's going to use the device to turn the stimulation off, and you'll see that he starts to develop some symptoms of Parkinson's. And you know, I'll warn you, it gets a little uncomfortable to watch because it's clearly not a kind of pleasant situation for him. I do assure you that he will turn the stimulation back on. So bear with that. Okay, so as I say, I will kind of narrate the video here so you can kind of hear what's going on.
This is the remote control.
Okay, so this is the remote control. This checks that I'm on. Okay, so the device is saying that the stimulation is currently on. Now he's going to turn it off. So you won't be able to read that, it's shaking too much.
I can't control this.
So he says, I can't control this. He's deliberately holding his arms out, but the shaking he can't control. So he says, I can shake some quality cocktails with my right hand. The left is the royal wave. His voice is gone. Harder for him to get words out and speak. He's completely rigid.
If I try to stand up, I'd fall.
If he tries to stand up, he'd fall.
If anyone thinks I'm faking this, I'm not.
If you think he's faking it, he's not.
Control the tremors and dyskinesia, the twisting in my neck is called dystonia and it is really quite uncomfortable.
So you saw his neck twist, that's called dystonia.
So as you can see, the tremors are getting much worse.
The tremors are getting much worse.
I'm in quite a bit of discomfort and I'm about to drop the remote control, which will be fun because I won't be able to turn myself back on.
So he says he's afraid he's going to drop the remote control, so he's going to turn the stimulation back on.
Almost automatically.
So
Power comes back on.
So with the power back on, he can, you know, move normally.
I thoroughly recommend it. It's life-changing.
So in case you couldn't hear that, he says, you know, if anyone is considering deep brain stimulation, he thoroughly recommends it. It's life-changing. So, you know, I've watched this video many times. It still is amazing to me how, you know, there's this procedure that seems to be so effective and almost magic. You know, as soon as he turns the stimulation off, the tremors start to kick in and they get worse and worse. And then when he turns the stimulation back on, suddenly he's kind of able to move normally. Okay, so the stimulation that was being used in, you know, in his treatment is something like this. I don't know, you know, the exact specific details, but this is kind of the standard deep brain stimulation treatment. There's electrical signal at a constant frequency, about 120 hertz, and it's a pulsatile stimulus. So it's kind of, you know, a short, big input. Here I drew it as a positive input. And then there's something they call charge balance. So the amount of current injected here and injected in this negative part balance each other. And so this is always on as long as, you know, the pacemaker is on. He can turn it on and off manually if he wants to. Now, what frequency, what stimulus would one use? Well, this has to be tuned by a neurologist. And it typically requires multiple sessions, and it may be necessary to retune as the disease progresses. But this is kind of the standard. And, you know, I can say that, you know, I've asked some of the people that do these, you know, that implant the electrodes and so on, you know, ask them why 120 hertz, and it's kind of like, well, you know, the first people who tried this, that's what they tried and it seemed to work. You know, so there's been, you know, definitely a lot of thought about how it works, But it turns out it's not really well understood how deep brain stimulation actually works. Okay, now what I've shown you is a video that pretty much demonstrates that deep brain stimulation does work. And so you might say, why are we still talking about this? So a big question is there a better way to do this? And what does this mean? Well, it can mean a couple different things. One is, can we do this using less power? So the pacemaker implanted in his chest has a battery, and the battery has to be replaced every 2 years or so. And to replace it, you have to have surgery. And of course, surgery always has risks associated with it. And so if you could use less power, the battery could last longer, and you may not need to replace it as often. Also, if you think about what's going on here, there's an electrical signal being injected into the brain tissue. It seems probably it would be prudent to use as small of a signal as possible. Of course, the signal is small enough that it's not somehow frying the brain tissue, but you might imagine that the signal kind of spills out a little bit into nearby brain regions. And if you use a smaller signal, there's less of that happening, and hopefully that means there's less side effect. And also, this in the control world, we would call this open loop control. And we know that there's a lot of advantages to something more, closed loop kind of feedback control. And so, maybe by thinking in those terms, one can come up with a better way to do deep brain stimulation. Okay, so with that as kind of the motivation, here's just a quick outline to give you an idea of what's coming. The first half of the talk will be on trying to control a population of neural oscillators, where we're trying to desynchronize the neural activity. The control methods here, I'm going to just focus on a few of these we've worked on: chaotic desynchronization, optimal phase resetting, and phase density control. And then the second half of the talk will be a kind of more detailed model for the basal ganglia neural circuitry. So the basal ganglia is the brain region that's associated with Parkinson's and motor control and things like that. This gets more into the data-driven aspects. And here the goal is going to be to reduce the so-called beta band activity. And the control method will be a data-driven machine learning approach. Okay, so the challenge for both of these halves of the talk will be that you really only have one input, but you're trying to control many neurons. So there is a single electrode implanted into the patient's brain. Well, technically, usually on each hemisphere, there's one electrode. But the point is you have one electrode trying to control probably at least 100,000 neurons, maybe even 1 million neurons. And that presents, of course, a huge challenge. It's severely underactuated. It's not possible to control each neuron individually. So I think what this means is one has to be a little bit clever in coming up with a way of controlling these. Okay, so I'm guessing that not all of you have done kind of mathematical neuroscience before, so I want to just give you a quick overview of how mathematical neuroscientists view neurons. So the biophysics is kind of captured in this picture here. The membrane of the neuron is a lipid bilayer that electrically acts as a capacitor. And then you have protein molecules that kind of go across the membrane, and they act as gates for ions to flow either in to out or out to in. And electrically, these act as nonlinear resistors. So Hodgkin and Huxley back in 1952 published a very, very influential paper on the neural circuitry associated with this membrane here. So this is, just, I mean, you could view it as a single neuron or even less than that. It's just kind of a patch on the axon of a neuron. And the way I like to think about this is, suppose you put an electrode inside the neuron, injected some current I, and you wanted to know how could that current move from the inside to the outside. Well, you could have a capacitive current associated with changes in the voltage So it turns out capacitive current is C, the capacitance, times dV dt. Then you can have different ions flowing across. You could have sodium ions, potassium ions, and here they call this the leak current. It's basically all the other ions that could be crossing the membrane. There are these nonlinear resistors associated with the ionic flow. And then these batteries you can think of as just ionic pumps that maintain a concentration difference between the inside and the outside. Okay, so we have an electrical circuit, and you can then write down some differential equations that describe the dynamics associated with this. Now, this first equation is essentially just Kirchhoff's law that says that the current coming into this node is equal to the sum of the currents leaving the node. Just rearranged a little bit. The C dV dt is the capacitive current I mentioned. So you have the injected current, you have a sodium current, potassium leak current, and these are called conductances, so they're like 1 over the resistance. But you'll notice there's these extra variables, N, M, and H. These are called gating variables, and they obey their own differential equations as well, with some functions here. And these functions have, somewhat complicated forms, and you have a lot of parameters. So this, you can view this as the equations for one neuron, and you see that, you have 4 differential equations coupled nonlinear with a huge number of parameters. It's a pretty complicated system. And this is just one. I should mention that this, the neurons that they were modeling were from a squid. They call this the squid giant axon. And at the time, this was used because it turns out the neuron is big by neuronal standards. And so it was easier for them to do the experiments that were needed to come up with these equations. And I want to mention these were not derived from first principles, although there's a lot of insight based on kind of, how a circuitry, how an electrical circuit would work. And one last comment, they won the Nobel Prize for this work, which I think was probably well-deserved, because in some ways this is like the hydrogen atom of neuroscience. Most modeling of neurons is based in some form on these equations. Okay, now if you were to simulate these equations, this is the type of voltage trace that you would find. So the voltage builds up slowly and then very rapidly spikes up and then drops down and builds up slowly and so on. And here I've chosen a parameter. This would be the injected current coming, say, from an electrode inside the neuron. And we get periodic spikes, or they're often called action potentials. Okay, so this is just to give you an idea of, the sort of model that I'll be talking about here in the first half of the talk. Now, the equations are, as I mentioned, they're somewhat complicated. So it's a system of 4 coupled nonlinear equations. I'm going to go through a reduction procedure that is quite useful for understanding the dynamics and also coming up with control algorithms. So imagine this equation dx dt equals f of x is my Hodgkin-Huxley equations. So I've just kind of abstracted it, or it could be if you're looking at neurons in another brain region, or for humans, you would have a different set of equations. But I'm going to assume that these equations have a stable periodic orbit. So this would correspond to the periodic spiking that I just showed. Now, we can define something called an isochron, which is a set of initial conditions that share the same so-called asymptotic convergence to a periodic orbit. And associated with the isochrons, you can view them as a level set of a phase variable theta, which is a function of the state of the system. And we parameterize theta such that on and off the periodic orbit, we have simple dynamics. d theta dt is just equal to 2 pi over the period, which we define to be omega. And here I have a little cartoon to illustrate what's going on with isochrons. So I'm plotting the voltage here and a gating variable on this axis. The black curve is the stable periodic orbit associated with spiking. So when V becomes large, that's when the neuron is spiking. And The blue curves are the so-called isochrons. And what I'm doing is I'm starting with 3 initial conditions on the same isochron, and it's a stable periodic orbit. So the trajectories all approach that stable periodic orbit. And you can see they approach in phase with each other. And so when these different neurons spike, they're all spiking at exactly the same time. And this is just, we call the phase circle. So, theta would be the, the phase variable that I'm talking about. And remember, d theta dt is just equal to a constant. So here it's, it's just moving around at constant speed. Okay, and oops, if we start with initial conditions corresponding to different isochrons, again, the trajectories all approach the periodic orbit, but they approach out of phase with each other because they live on different isochrons. So you can see as far as the spike time is concerned, these 3 neurons are spiking at different times. Okay, so isochrons are related to something called phase response curves. And a phase response curve is defined, I like to look at this definition here. You can imagine, suppose you did a voltage perturbation to your neuron. So you let V go to V plus delta V. Associated with that perturbation can be a phase change, a delta theta. So kind of going back to this here, if I let V go to V plus delta V, I've kicked myself to a different isochron that has a different theta value. Remember, isochrons are level sets of this theta variable. And so I have a delta theta associated with that. And then I take the ratio of delta theta divided by delta V, take this limit as delta V goes to 0, and that gives me this so-called phase response curve. And the phase response curve captures the effect of impulsive perturbations in the voltage. This is the phase response curve associated with the Hodgkin-Huxley equations. And what I think is really cool about this is if I do the same exact perturbation at different phases of the periodic orbit, I get a different result. So if I do a positive delta V perturbation at this phase of the periodic orbit, it actually kicks myself to a lower phase. It makes it take longer for the neuron to fire. Whereas if I do the same exact perturbation here, it kicks it to a higher phase and it makes it fire more quickly. So a lot of our control algorithms based on this phase response curve are kind of taking advantage of that the timing of a stimulus matters. Now I want to emphasize that Z, this phase response curve, can be measured experimentally. And so in some sense, and this is maybe not what you imagined when you heard this was going to be a data-driven approach, but this is a lot of the reason why we developed control algorithms based on the phase response curve, because it can be measured experimentally. Not saying that it's easy, so you have to get a neuron, a living neuron in a petri dish, and do something called patch clamping, and make measurements before the neuron terminates, passes away. But in principle, it can be done. And so, as I say, this is a form of, let's use data in a smart way in order to, generate something that can be used to do control. Okay, so I'm going to go through a couple control algorithms based on this phase response curve. And then, as I say, I'll talk a little bit about this kind of more machine learning approach. Okay, so this first one I'm going to talk about is called optimal chaotic desynchronization. So I told you about this phase reduction, d theta dt equals omega. If you have an input, something like a deep brain stimulation input, the way it comes in is like this. So theta I would be the phase of the ith neuron. And I'm imagining I just have 2 neurons here to derive the idea. Both of them have the same frequency and the same phase response curve, and they're both feeling the same input. Okay, so this is how, when you do the phase reduction, you account for the inputs, phase response curve. If we let phi be the phase difference between these 2 neurons and do a Taylor expansion, we get d phi dt is given by this expression. And if I get rid of this nonlinear term, then I can say that phi goes as e to the capital lambda times t, where capital lambda is a finite time Lyapunov exponent that depends on the derivative of the phase response curve and the input. Now, for those of you who have studied chaos, you know that a positive Lyapunov exponent is associated with chaos. And what it means is that you have this exponential divergence of nearby trajectories. So here, what we're going to do is an optimal control where we're trying to make this Lyapunov exponent as large as possible, okay, positive and as large as possible, so that the phases of the neurons exponentially move away from each other. And remember, there's this hypothesis that some of the symptoms of Parkinson's are associated with pathological synchronization. That means all the neurons have phases close to each other. Here, with this control, it's going to move those phases apart, so it corresponds to desynchronization. Okay, so I'm going to skip the details of the optimal control. I'll just show the results. Now, here I'm showing some simulations with no control, just to kind of give you an idea of, you know, what the behavior would be. So here I'm simulating 100 neurons. They're coupled. The coupling is trying to bring them all in phase with each other. They're also noisy, which is trying to spread those phases apart. There's no control here, and the neurons are spiking when they cross over here, okay? And I should mention, I don't think I mentioned, you know, the way we parameterize the phase variable, theta equals 0 would correspond to this maximum voltage. Okay, so that's in the absence of control. Here I'm starting with the neurons fully synchronized with each other, but using this optimal chaotic desynchronization. When the dots are red, that means that the control is on. When the dots are blue, we've decided that the system is desynchronized enough that we don't need any stimulation. And so you can sort of see that, you know, the dots are crossing through this maximum voltage, you know, separated from each other. So they are in fact desynchronized. So here, you know, as I say, we judge that they're desynchronized enough, so we turn off the stimulation. But then when they become too resynchronized due to the coupling, then we turn it back on. Okay, so, and it turns out that, you know, what we're optimizing is the Lyapunov exponent, but we're also trying to minimize the amount of power that's needed to do this. And this approach uses at least a factor of 100 and possibly as much as a factor of 1,000 less power than kind of the constant frequency pulsatile deep brain stimulation that's normally used. Okay, so another method that we've developed is again inspired by these isochrons. So remember I showed you a plot of the isochrons. It turns out that if you follow them, they spiral into an unstable fixed point for the system. And this we call a phaseless set because you cannot define the phase at that point. And the control method we used here was we call optimal phase resetting. This was inspired by Art Winfrey, but he did it not so much as an optimal control problem. The idea is, suppose your neurons are all kind of in phase with each other. Let's design a control that brings us into this neighborhood of the phaseless set. Then the system is very sensitive to noise at that point because you could imagine, you know, if every neuron is feeling a different realization of noise, it gets kicked to a different isochron. And then when the stimulation gets turned off and you return to the periodic orbit, they return out of phase with each other, and therefore the system is desynchronized. So again, you can formulate this as an optimal control problem. Here I'll just show a movie. So when the dots are red, that's when the control is on. Remember, you know, every neuron is feeling the same control. There's only one electrode that you have here. And so this control method is basically trying to steer all of them into this region of phase space where you're very sensitive to noise. And as you can see, the dots nicely separate from each other, and that's, you know, desynchronized. Okay. The last method I want to show you based on the phase response curve is what I'll call phase density control. So you might imagine if you take the number of neurons to infinity, you can come up with a PDE, which describes the density of the phases for your system. And then what we did was we formulated a control acting kind of directly on that phase density. Here, just to orient you, the blue curve I'm imagining is the current phase density for the neurons. So, theta equals 0 corresponds to the neurons firing. And then the black curve is our target distribution. And we're going to design a control. It'll be shown here. And then this is the L2 difference between where we are, the blue curve, and where we want to be, the black curve. Okay, so here, given this control, we're able to drive this blue curve towards the black curve. And, the longer you wait, the closer you get. Okay, so here we started with a synchronized system where there was kind of this hump, which means the neurons are all kind of in phase with each other. And we're targeting this final distribution where it's flat, and this would correspond to desynchronization. And one nice thing about this method is you can use other target final distributions. So here I'm kind of doing the reverse. I start with a completely desynchronized distribution, and I'm forming, we call this clustered state, where half the neurons are in one cluster, half the neurons are in another cluster. So there may be some advantages to steering your system towards that sort of state rather than a fully desynchronized state. And here I'm just showing if I have a population of oscillators feeling this signal, you can see that indeed they do separate into these two clusters. Okay, so all of these methods that I've shown you are based on the phase response curve, which I said is experimentally measurable. So you might say, hey, the problem's solved. We can stop the talk early. We're only halfway through. And I'll tell you, no, sorry, the problem is not solved. So we were excited by these results, and we've published papers on these, and my students have gotten publications, and a few of them have won best dissertation awards, and it's all very exciting, right? So we were thinking we want to apply this to a real system. And so I got in touch with Philip Starr, who's up at UC San Francisco. He actually does these surgeries, and he's also, I think his PhD is in chemistry, so he's also very well versed in modeling and things like that. And so I said, hey, can I tell you about the work I've been doing, and it'd be cool if some of this could be translated to real patients. And so I kind of gave him a talk like what I just gave you so far. And he, politely kind of smiled and said, yeah, that's very interesting work. And they said, but there's no way that any of this would work for a real patient, because it just assumes too much information and it's too idealized. So, we're considering identical neurons, so no heterogeneity. Sometimes we're considering systems without noise. And of course, the brain is a very noisy place. So, and it's just much more complicated. There's neurons interacting with other neurons, different brain regions, and so on. So what can you actually measure in a human patient? You can't measure phase distributions, unfortunately, although we have some ideas for how you might be able to extract out the phase response curve from some kind of population level measures, but this is more honestly what you might be able to measure. And even this is not trivial for a human patient. So this is something called the local field potential. And it is essentially you stick an electrode into the brain region of interest, and you just measure whatever electrical activity can be detected. So it's some averaged, filtered activity of whatever neurons happen to be close to the electrode. And here I'm showing two different traces of this LFP, one for a healthy patient, one for a patient with Parkinson's. You might be able to say, well, it looks like some oscillations are bigger. But I can assure you we could find a different, section of this time trace where things would look almost flipped from each other. And so it is, in fact, very difficult by eye to tell the difference between a healthy patient and a patient with Parkinson's disease. Okay. Now, these traces, it turns out these are not from real patients, although they do capture the dynamics well for real patients. These are coming from a more realistic model than the models that I've shown you up to this point. So up to this point, the models have kind of been a single population of neurons that are all identical and so on. Here, this is more realistically what's going on. So you have the different brain regions. So this box is the basal ganglia. This box is the thalamus. Then you have the cortex. And you have projections from, say, the cortex to the subthalamic nucleus. Then the subthalamic nucleus projects onto the globus pallidus internus and so on. And you see it's quite complicated, right? So this model here, and we're using the implementation from this paper here, much more accurately is capturing the dynamics associated with Parkinson's disease. And to orient you, the stimulation goes into the subthalamic nucleus. So this is the deep brain stimulation would go here. And the measurements of the local field potential are in this GPI region. Okay, so what we were interested in doing is, taking this model, which is much more realistic, and it outputs something that is like what you could actually measure for a human patient. Can we develop a control algorithm which now does something useful? And here we're going to be looking at the power spectrum of the LFP, and in particular the beta band activity. And I'll say a little more about this. And you may recall that there's some evidence and a hypothesis that Parkinsonian symptoms are associated with elevated beta band activity. So what we want to do is design a control that brings that beta band activity down. Okay, so here's our, kind of big picture, our control strategy. We call this adaptive deep brain stimulation. So there are bursts of beta band activity. The beta band is, you've maybe heard of alpha waves and beta and gamma and theta and so on. So the beta band is roughly 15 to 35 hertz. The bursts of this activity seem to be associated with the symptoms. What we want to do is a short-term prediction of whether or not one of these beta band bursts will occur in the next time window. And then based on that information, adjust the frequency of the stimulation. Okay, so we're going to be using the same type of pulsatile stimulation as what I showed you right after the video of the guy from YouTube. But instead of using a constant frequency, we're going to adjust it as needed according to this prediction. And what are the advantages over constant frequency DBS? Well, it can save power. So you're only using the level of stimulation that's necessary. We believe that it could reduce side effects because, again, you're only using the stimulation that's necessary. So one might expect that lower frequency stimulation is less likely to cause side effects than higher frequency. And also, well, we want to adapt to the status of medication. So a typical patient will be on medication, and also some of them will be using deep brain stimulation as well. And of course, the medication can kick in and wear off, and that will affect the dynamics of the neural circuitry, and therefore maybe you want to adjust the frequency of the DBS in order to do this. So we scratched our head a little bit, looking back at this model and saying, well, this is pretty complicated. I don't know if we can do a phase reduction of this to use the techniques we've done so far. So that brought us to machine learning. And this is my somewhat humorous, somewhat cynical view of machine learning, where you have an input that goes into some black box here, and then suddenly you get this wonderful output, which somehow takes a signal where you can't say much about it and splits it into something which maybe you can say something useful about. Now, I should say, I of course appreciate the power of machine learning, and I'll be honest, probably many of you listening to me right now are more experts in machine learning than I am. So I'll try to do my best to convey the approach that we've been taking. So we're trying to, as I say, do this realistically, where what you could measure is this local field potential. So we're taking as input this LFP, and more precisely, we're taking the power spectrum of the LFP, feeding it into an encoder. So this is our autoencoder. Feed it into an encoder to develop some encoded variables. And then to determine these encoded variables, we then have a decoder and reconstruct it and train it. And then once we've trained it well, we cut things off and then just look at these encoded variables. And the hope is this gives us a lower dimensional representation of the LFP that we can then use to make predictions. Okay, so just to give you an idea, so these encoded variables are, again, the power spectral density over some frequency range. Here, I'm just trying to show you, for some different inputs of the power spectral density, the red corresponds to the lower values of the encoded variables. So encoded variables 0, 1, and 2. We found 3 encoded variables work pretty well. So, what exactly are they capturing? Well, you can see they're capturing some elements of the shape of this power spectral density. Okay, so now we're going to do supervised learning of this adaptive deep brain simulation. So we're going to train a deep neural network with inputs being the encoded variables for the power spectral density over a 2-second interval. And then we're also inputting some candidate control input frequencies. And the output from this neural network are going to be the best stimulation frequency to keep that power spectral density below threshold for a desired fraction Q of the time for the next 2-second interval. So remember, this beta band activity, if it's elevated, it's associated with Parkinson's. What we're trying to do is get that power spectral density below some threshold, at least maybe half the time for the next 2-second interval, or this Q will be a parameter that we can change and train our network in order to do. So to give you an idea that this looks promising, so here, and I should say, we start our neural network with just random initialization. So it's learning on the fly. And here we're targeting that we're below threshold 0.5 of that upcoming window. And we're just doing 2 different trials here. And you can see that the frequencies do change quite a bit, and they change differently for the different trials, but we're able to hold things pretty well around this 0.5 level. Okay, and as I said, Q is something that we can tune for our network. So these are all, again, cases where we're starting with just a random initialization for our network. And for different values of Q, so here, this would be the 0.25, and I should say the thin lines are individual realizations, and the thick line is kind of an average over those realizations. So when we're targeting 0.25, we can do well. When we're targeting 0.4, 0.5, 0.6, 0.75, and then these are the frequencies that are necessary in order to bring you there. And you see that, okay, you might have to adjust the frequency quite a bit in order to hold yourself at that level. But what we're imagining is that one could use this Q as an extra parameter to kind of say, how carefully do we want to control for this patient the level of beta band activity above threshold. And so using this algorithm, you could then, given that Q, adjust the frequency accordingly. And here's one thing that we think is very cool. So when a patient is on medication, you would expect the parameters associated with their neural circuitry would vary. And this is kind of a busy plot, but we're just choosing different frequencies of variation of the parameters. And so basically in the model here, we're varying the patient from being healthy to having Parkinson's disease in a sinusoidal fashion over some time intervals. So this is time in seconds. And then this control algorithm that we've developed will adapt because it's always taking in the new information and adjusting the weights in order to make that happen. So here we're trying to do this moving probability, the Q value at 0.6. And it's just showing for all these different periods of variation, it's possible to choose the frequencies, for the next time interval in order to keep yourself very close to that. And this is something when we talked to Phil Starr about what they felt, would be a nice target for this adaptive deep brain stimulation. They felt it would be, very good to have something that could adjust to this medication level. Of course, we're not making an amendment of the medication. It's just based on kind of the dynamics of the model and the deep neural network learning what level of stimulation or what frequency of stimulation to use to make this happen. Okay, so we feel good about how this is happening with the model. And so the next step is to apply this to human data. And so Phil Starr's group was very generous in sharing some data with us. And we don't have this done yet, but I'm excited to show you some kind of new results that we have with the human data that we think is really pointing in a good direction. So the data here is LFP recordings from humans. The recordings tend to be about 1 to 2 minutes long. They're all recordings for a single patient, but recordings over different trials over several years. And there's data for the DBS stimulation on or off and medication on or off. And so there's 4 different combinations. You could have DBS on, medication on, DBS on, medication off, and the other 2 combinations. So it's not a lot of data in all honesty. So, 22 1-minute trials all for 1 patient. So we wanted to see, what can we extract from this? So we've tried a couple different methods. The thing that seems to be working well now is regression. So this is kind of a neural network style regression. So the inputs are encoded variables. So I showed you these encoded variables before, and actually I didn't mention this deliberately, but these encoded variables actually are from the human patient data. And we use the same encoded variables for the model. So we feel pretty good about using real data encoded variables, and the model still works well. But so here for our regression problem on the real data, we input the encoded variables. Then this is similar to what we did for the control, but a little different. So we're essentially looking at a sliding window inside the current interval. So let's say the current interval is 1 second. We look at a sliding window of 100 milliseconds, and we slide through that whole interval, and we look for the maximum proportion of time that the peak of beta activity is above a threshold inside that current interval. Okay, so this is, we believe, a nice way of characterizing if there's a burst of beta band activity. And then for our regression, we also input, is the DBS stimulation on or off, or is the medication on or off? And the output is going to be the maximum proportion of time that the peak of the beta activity is above threshold in that sliding window. So basically, we're taking what's happening in the current interval and predicting what's happening in the next interval. And then the idea will be, we'll adjust the frequency of stimulation based on that. Okay, so does this work? So here we're doing a comparison between a no change hypothesis. This would basically be just assuming whatever happened in the current interval is going to happen in the next interval. And it turns out when you look at the data, that's a very good prediction. It turns out things don't change a whole lot. Every once in a while, you get this burst of beta band activity. These, oh, did I mislabel these? The dashed, I mislabeled these, sorry. The solid is or no, this is correct. I apologize. This is hot off the presses here. The solid is from the regression. The dashed is from the no change hypothesis. We're plotting the error here, and you see that using the regression is better. And the way to interpret these lines, so for example, this curve here corresponding to the third quartile absolute error, 75% of the data has error below this line. So you can see that the regression is giving a pretty significant smaller amount of error than the no change hypothesis. And we're also considering other things kind of based on PCA and so on. I don't have the results to show you just yet, but we believe that this regression algorithm is giving nice results. And if we look at how is it improving things? So here I'm plotting in the current window the max average bursting activity. So if this was 0, that means in the current window there's no bursting activity. If it's 1 in the current window, it's all above threshold. So there's kind of a beta band burst. And then the mean absolute error in our prediction, well, and over here, this would be for the no change hypothesis. Maybe it's easier to start here. So if there was no beta band activity in the current window and in the next window, there was again no beta band activity, then there's no error associated with the no change hypothesis. But if it turns out that there was bursting activity over that entire interval, then you would have an error of 1. And then you have all kinds of errors in between. You can see sometimes in the current window there's maybe 0.4% above threshold, and then the mean absolute error, well, if in the no change hypothesis you say it'll be 0.4 again, but in fact there's 0, then you would have this 0.4 absolute error. So if you look, our neural network regression approach gets rid of kind of those X's. So it makes much better predictions than the no change hypothesis if there's a level of activity above threshold between 0 and 1. And it also does better up here. So it's driving these, if you look at the color scale, the no change hypothesis, you have more realizations up here. Here, when it's green, basically you're pushing the error down here. Okay, so at any rate, we feel pretty good that this regression is giving us less error. This, as I say, is very hot off the presses. The graduate student, Tim, who was doing this, sent me these figures, I think, certainly within the last week and probably within the last few days. So what are our next steps here? Well, we already have some data for more patients. We're interested in applying these ideas to those to see if we can get comparable level of improvement in making predictions. We would also love to have, and this is very difficult, we would love to have patient data for different stimulation frequencies. So I told you that some of the data is for stimulation on. This would be for constant frequency DBS. For training our algorithms, it would be great to have patient data for different stimulation frequencies. If that's not available, then we would do the training using the models such as I showed you. And of course, one of the big questions is how to implement this for real patients. So we're in touch with our collaborators at UCSF, and we're very excited to see where we will be able to take these ideas. Okay, so just to wrap up, to summarize what I've told you about, so this is all motivated by Parkinson's disease, deep brain stimulation. Our goal, well, in the first half, it was to desynchronize the populations of neural oscillators. In the second half, it was to reduce beta band activity. These are related to each other, certainly. I talked about the control based on phase models, which I will emphasize again. In some sense, this is data-driven because it's possible to measure these phase response curves. So I told you about chaotic desynchronization, optimal phase resetting, phase density control. And then the newer work, the data-driven machine learning approach to adaptive DBS using our autoencoder, deep neural network, regression also when we're doing the real patient data. One thing we're very excited about is this algorithm adapts to slowly changing parameters. So we feel that this could be useful for situations where a patient is on medication and DBS. How can we, adapt to what they need based on whatever their current state is? Okay, I'll mention, just very quickly, we've been thinking about this problem for a while. We have other ideas for how you can control. This is more based on the phase sorts of models, but we also did some supervised learning for binary control, some reinforcement learning, and then other types of related research. And I'll just mention, we also have other experimental collaborators, Tay Nedoff's group and Ken Showalter's group, where we've applied some of these ideas to chemical oscillators, also controlling circadian rhythms. If you like this phase sort of stuff, here's a tutorial we wrote recently that kind of talks about how the phase reduction works and how you can develop control algorithms based on that. Perhaps the more important slide is this one, which is the collaborators. So of course I didn't do all this by myself. The work on kind of the machine learning adaptive DBS was done by Tim Machen. Some of the other work on the phase control was done by Dan Barat and Ali over the years. Per was another student who kind of started with this. He was kind of the brave first mechanical engineering student to work on neuroscience applications in my group. Here are the collaborators from UC San Francisco. We've had high school students working on this. My experimental collaborators at Minnesota and West Virginia, and some other people who have been useful for different aspects of this. And of course, thanks to the NSF for funding this work. And I'll end that here with the mad scientist slide here is my attempt at brain control. So with that, I thank you for your attention, and I'm happy to answer any questions that people may have. So I will stop sharing. So maybe you can see me, and I'm not sure how you do questions. If you want to let people ask me or whatever, I'm totally open.
What we've been doing is have people type them in the chat, and then I would convey them to you.
Okay.
So yeah, feel free to type your questions in the chat. As well, we're waiting for people to type out their questions. I have one. So this is more on the mathematical side, just based on my interests, and about the phase density control. To what extent have you thought about coupling, including coupling in that problem?
So we have thought about that, and actually there's a group in Iran who got in touch with me and wanted to study that problem. So we've been thinking about it a little bit. I've been too busy to kind of be giving them the feedback they deserve, but that's something that's kind of next on my radar, wanting to work on that. And so just to give kind of an idea of how we want to do that. So we want to still do phase models, but incorporate, and you may know, so for phase models, you can get phase difference coupling when you kind of average the system. And so we're kind of interested in looking at systems that have that coupling and also have, these external inputs. And, we believe that we have, and noise, so why not throw noise in as well?
Sure.
So we believe we have some ideas for how that can be done, but, that certainly I think is worth exploring further, because the real systems will have coupling. And, often the way we've derived our control algorithms is we assume there's no coupling, and then we develop the algorithm, and then we try it numerically on the system with coupling and verify that it works. But we've only done a few cases where the coupling was actually included in the development of the control algorithm. And I think that's a very ripe direction, but it's also a very challenging direction.
Yeah, gotcha. Thanks. Okay, so we've got several questions in the chat. I imagine there would be some power costs to actively monitor brain signals and adapt the applied signal power in response to that. Given that, how much power can you expect to save from adaptive brain simulation?
Yeah, that's a great question. And in fact, our collaborators at UCSF have asked us that same exact question. And we don't quite know the answer to that yet. But I think that, certainly there would be a power cost associated with that. I think it's even possible that it would use more power than, just running the constant frequency stimulation. But though that's not the only reason why we think adaptive DBS might be useful. We think that, adapting to the medication state could be a very beneficial thing. And also just, there may be, and a lot of this needs to be explored, but there's a sense maybe that higher frequency stimulation is more likely to lead to some side effects. And so maybe the higher frequency stimulation, you're willing to put up with side effects because it's needed, because you're trying to suppress this beta band burst. But then when it's not needed, you're doing lower frequency stimulation where there's less of a chance of the side effects being important. And so you might be willing to use more power, even though, it's like on average you're using more power, but you're only using it at the times when you really need it. And I should mention that, Phil Starr's group, part of the reason why I got in touch with them, they have another form of adaptive DBS that is currently, being used in some human patients. I don't think it's being used kind of out in the field, so to speak, but where it's more of an on or off. So sometimes they turn the DBS completely off when it seems the patients don't need it. And, again, there could be similar advantages to what I just sketched out for that. But that's a great question.
Cool, thanks. Next question, are there approaches to control of unstable periodic orbits in neural models? Do the unstable periodic orbits appear in neurodegenerative diseases?
Yeah, that's another very good question. And the answer is yes. I've seen some, kind of chaos control sorts of algorithms applied to neural models. I'm not sure if they're important for neurodegenerative diseases or not. They may be, but so there could be, for example, a separatrix of sorts between different states, and the unstable periodic orbit is separating those. Some of the work we've done has been kind of control based on those sorts of ideas. I should also mention, we've worked on cardiac systems, and some of our cardiac control is trying to control the system towards an unstable periodic orbit. So there's a phenomenon called alternans. So you also have action potentials for heart cells, and for a healthy patient, you would have all of these action potential durations, they're called, being roughly the same for subsequent heartbeats. For alternans, it turns out these durations alternate between short and long, and this can lead to bigger problems, bigger arrhythmias like fibrillation. And so what we've done, it turns out there's, for the dynamical systems people, there's a period doubling bifurcation where the period one solution becomes unstable, and then the period two solution becomes the stable alternate solution. And so there, what we're trying to do is control to the unstable periodic orbit, which corresponds to the healthy state. And so we have developed an algorithm for that. It's maybe a little different from what you might expect, because here the periodicity only comes from pacemaker cells, not an implanted pacemaker, but there are cells that are firing periodically, and they then kick the excitable cells and make them fire periodically. And then that's where you get the period doubling bifurcation with that. But yeah, I think, I'm very excited by these approaches that you think about kind of from a dynamical systems perspective, what's going on. Like, are there unstable periodic orbits? Are there, are you trying to change the frequency of the periodic, whatever, things like this. And can you then develop control algorithms that kind of exploit that insight from dynamical systems? So yeah, that's a great question. And I think I saw my friend Predrag Svetanovich pop up. I don't know if he's there or not, but he's worked a lot on unstable periodic orbits. And you know, I think the interplay between unstable periodic orbits, control, that's something that's very interesting and probably worth investigating more.
Cool, thanks. So there's 2 more questions in the chat right now.
Sure.
Did you try for the data-driven and the prediction any type of Kalman filtering, such as the UKF?
Yeah, so UKF, I assume, an unscented Kalman filter. We have not tried that, but there is another great question. I particularly like, there's this work by Steve Schiff and Tim Sauer, I forget who else was involved, but on, you know, applying an unscented Kalman filter to kind of fit a nonlinear model to data. I think they were using numerically generated data, not patient data. But I was very impressed by that work. And I think what that's doing is, you know, it's kind of a system ID approach. I'm not sure, maybe this is what the person asking the question is, or maybe not, but my interpretation, it's kind of a nice system identification approach where you're assuming a certain form of the equations, kind of a Fitzhugh-Nagumo style model. And then the unscented Kalman filter is just looking at the voltage trace and figuring out what the correct parameters would be in order to you know, get a good model, which then could be used for prediction. We have not tried that. I think that's a great idea. And you know, maybe one of my next students, that would be a good project for them to think about. And I should say, you know, there's a huge number of approaches out there. I'm currently reading the nice book by Steve and Nathan on data-driven dynamical systems, and I'm learning a lot from that. You know, some of these things I've looked at before, and some of them I haven't. And you know, I think that, you know, it's an exciting time to be able to you know, think about all these different approaches and what would be the best one for the particular problem at hand.
Great. Yeah, so the final question we have in the chat here is, are you envisioning closed loop control strategies that incorporate sensing of Parkinson's disease symptoms like hand shakiness? And are there available data on that?
Yes. We are not actively doing that, but that's another great idea. There's a group at Oxford that I think has done that a little bit. And, you know, so you could mount an accelerometer on a patient's finger, for example, and then you have, you know, a measurement of how the tremor is. And then based on that, you could, you know, presumably adjust the frequency or turn the stimulation on or off. I think that, you know, what they were doing, I believe they were actually measuring phase response curves based on accelerometer data, which is very cool. I don't think they were kind of closing the loop and doing it for feedback control, but I can't say for sure. I think that's a great idea. One thing I would point out, though, is that tremor is only one symptom, and often patients, I'm told, find the rigidity to be the most frustrating symptom. The fact that, you know, they're not able to initiate movement in the first place. And I'm not sure, I mean, there might be other ways of detecting rigidity using, you know, accelerometers or, you know, other, who knows, maybe EEG signals or something like that that could be used. I think this is a wide open field, though. I mean, we're just scratching the surface at ideas of what could be done. I think especially now that these machine learning techniques are coming on board, you know, you could perhaps, I mean, I just saw, you know, there was, this was on Facebook, so, but I think it's probably true. A group at MIT was looking at people's coughing and, you know, being able to detect, is that cough associated with asymptomatic COVID or just a normal cough, you know, using a machine learning algorithm. And, I've heard of, analyzing people's speech, and being able to detect if there's maybe some disorder for that person. And some of this, you always have to take with a grain of salt. And is it being overhyped, or are you just fitting to, the training data, and then you try it on new data and it's totally different, so one has to, of course, be very careful about these sorts of things. But I think, we're in that data era now where there's a huge amount of data available. Can we sense something useful from that data? What we're doing in what I presented today is, from the LFP data, can you, at least extract out some information that allows you to make a short-term prediction about what's going to happen, and then do your control accordingly. Maybe somebody can think of other type of data that would give you something, either, comparable or even better. Maybe that comes from an accelerometer mounted someone's finger. Maybe that's an analysis of their breathing pattern, or an analysis of their speech pattern, or something like that, and then design control based on that. So this is all wide open, and hopefully, we can, as a community, work together to, bring better quality of life to people that have these disorders. So, yeah, I'd love to hear if you have ideas of how to do these things. I'd love to hear about them.
Great. Okay, well, I think that's all of the questions we have. Thank you so much for being with us and sharing your research. It was a great talk.
Thank you for inviting me. That was very nice. I don't consider myself to be a hardcore machine learning person, so I appreciate that you gave me the chance to talk to this audience.
Great. Okay, and we'll see everybody in 2 weeks.
Great.
Okay. Bye-bye.
All right. Thank you.